wanna make some speech end of section, end of day, but I will talk a bit because we start and I want to say something extra. First of all, this project it was very fruitful because of our team, every university's team is perfect and expert about their work, and with this teams, all teams take together, and this project results. We collect this project results, and thank you very much. Sure, we will continue, or we already discussed separately, but we will do extra further project. Erasmus key 2 or key 3, it can also cost, it can be. Especially I will focus about after project. I will focus and coordinate, try to coordinate all partners plus extra partners. We will try new project too. I hope our friends also wish to continuously work, because science not stop, always go in front. Thank you very much. Today's our speech chair is Professor Rodriguez. Please, your talk.
Good evening, everyone. Today we start with a presentation in sexism. Effect of essential oil isolated perforatum against Oturum species. Conducted by Professor Dr. Temalı Uluslararası, which he was born in Ruscus in 1968. He completed his undergraduate education in the Department of Plant Protection at Temalı University, Faculty of Agriculture. He completed also his doctorate in the Department of Forestry Engineering and Technical University in Trabzon. He has been a faculty member of Artvin Çoruh Üniversitesi Faculty of Forestry for 27 years. He is married and has 2 children. Please, Professor.
Dear Chairman and dear guests, good morning everybody again. Welcome today. I will talk about Bitkilerde and Hypericum perforatum essential oil. My presentation name Insecticidal effects of essential oil, isolated Hypericum perforatum against Bitkilerde. Forests are the large search of the world. They not only clean the air of the region where they are located, but also provide abundant rain. These natural researchers have got some problem, which is fire, diseases, pests, and human activities. Forest fires destroyed forests is one of the important factors in the world. Another one, bark beetles damage, is known as smokeless fire. Everyone say this damaging of bark beetles. Pests cause more tree damage worldwide than wind fires. In recent years, many insect species have caused damage to agricultural and forest areas in Turkey. The Eastern Black Sea region is also under the threat of these harmful insects. Pseudalima perspectalis is an important damaging insect in Black Sea region of Turkey. Boxwood moth larvae cause damage to boxwood trees. Pseudalima perspectalis has got 2 and 3 generations. One generation April-May, second generation June and August. Pseudalima perspectalis has present a particular series trait to fox trees. The young larvae consume the inferior epidermis and mesophyll of the leaf. The mature larva attacks the bark on the branches. In case of powerful attacks, the plants can be partially or totally destroyed. Insecticides are widely used in the control of the boxwood butterfly. Since a completed IPM strategy has not been developed. As a result of the use of synthetic insecticides against agricultural and forest pests, insects have become resistant and living things have been toxically affected. Due to the negative effect of the insecticides, natural enemies have begun to decrease, and this has made it necessary to research alternative substances in pest control. Today, as an alternative to synthetic pesticide essential oil, plants have begun to use it as pesticides. The plant Hypericum perforatum has essential oil that kills insects. In this study, the lethal effect of essential oil obtained from Hypericum perforatum plants on the larva of Pseudalima perspectalis were investigated under laboratory conditions. The larvae used in the study were collected from the boxwood in Hatila National Park and brought to laboratory with plant materials. Hypericum perforatum was collected from Artvin during their flowering period in 2023. The collected plant materials were dried in the shade and were cut into small pieces with the help of milk. The essential oil obtained in the Clevenger device within 3 or 4 hours were purified from water with anhydrous sodium sulfate. Essential oil were prepared in 10, 15, and 20 milliliter petrol solution at the rate of 1.2 for use in application. 1, 2nd, 3rd, 4th, and 15th star larvae of Pseudalima perfectalis were placed in plastical storage containers for toxicity testing. Sufficient amounts of fresh food leaves were placed for their feeding, and 1 milliliter of the stock solution were sprayed of them. As a control, pour waters plus ethanol was used for each larva stage, and nimazole was used as a positive control. Experiments were carried out under laboratory conditions, and each trial was carried out with 3 replications for each larval stage. The larvae were condensed at 1, 2nd, 3rd, and 4th days after the application in order to determine whether there is statistically significant difference between the result obtained was applied using the SPSS software HK. Hypericum perforatum essential oil tested against boxwood butterfly on star larva was compared in terms of toxicity at all times and all doses after applications, and heat were observed. In experiments, mortality rate is between 44.7% and 100% were observed in Pseudomonas perfectalis larva at all instar larva. At all doses, when mortality rates are examined, the highest mortality was observed in first and second instar larva. After 4 days later, at the lowest mortality was observed in 5 instar larva after 1 day later. When the mortality rates were compared according to application doses, it was observed that the lowest mortality rate for all larva stage of buxwood butterfly was 10 milliliter per and the highest mortality rate was 20 milliliter per. In the test, it was observed that 1, 2, and 3 stage larvae died on control hours in positive control. The effect of nemazal application 4 and 5 instar larvae resulted in mortality rates ranging from 72.2% to 100%, depending on the larva development stage, waiting period, and application dose. In addition, there was no mortality in larvae in the negative control for all applications. In the analysis of lethal dose toxicities, differences were determined between the doses. According to LD 50 and LD, lethal dose is 80 volumes. The highest toxicities were recorded in 1 star larva. After 86 hours, the least harmful amounts according to lethal dose 20 and lethal dose 80 volume were found in 5 instar larva as 9.37 and 14.11 milliliter larva. In the future, the use of plants essential oil as biopesticide against harmful insects will be more common. Based on these results, we can say that the essential oil isolated from Hypericum preparatum can be an alternative method for controlling the 1 and 2nd instar larva of the buxwood butterfly, which is one of the important pests of buxwood trees. In addition, this laboratory-based research should be conducted in the field and the results should be analyzed comparatively. Thank you for your attention. That's all.
Presentation, nice videos also showing the problem, problem with other insects, and so we also have some evidence of the efficacy of other essential oil. Mainly we are using cannabis oil that grows very fast, and so we can achieve it in big amount. So the point is always the cost of these applications that are effective. In the same time, if you have a big area, so which is your view of the real applicability in the agriculture? Because what we have as experience is mainly in flower, so in closed environment in which you have some pests and some insects, so it's relatively easy due to the limited volume.
Yes.
In open, it will be.
My idea is essential oil can be used in small area. Part of your gardens maybe is storage in home, because is much insecticide very effective for humanity in home, because it's a big problem for your health. But is essential oil no any effect and very good smelling. That is we can use in home is essential oil.
Thank you.
Thank you so much, everybody.
Thank you.
you.
Professor Bizi is Doctor of Biology, Associate Professor of the Department of Biology, Faculty of Natural Science and Health at Oturum Türleri State University. She has 35 years of experience at this university, where she gives lectures and practical courses in ecology and biology in undergraduate, graduate, and doctoral educational programs. Her research field is interdisciplinary. It deals with pollution of the modern environment, air, soil, water, biosphere, food products pollution, and their impacts on human health. She has published 1 monograph and more than 70 scientific papers on this topic. Professor Bizi takes an activity part in international and national scientific forum. She is the organizer, chairman of many international conferences and symposia, summer school and scientific seminars, as well as editor of various scientific journals and work collection. She is an academician of the Georgian Academy of Ecological Science and head of the Regional Ecological Service. Her research fields of interest: ecology, environmental management, environmental protection, plant ecology, soil ecology, pest monitoring, and disease.
Thank you very much. Yeah. So, hello, good morning. So, we are from Georgia, from Tıbbi State University, and we are friends for the biology department, Tıbbi State University, and I am very happy to be here today with you. And I would like to say special thanks to the organizers of this conference, Mustafa Kemal Gumuş and Hakan Gültekin. Special thanks. And thank you very much for our invitation, friendship, cooperation, and there is a special group working in this direction of medicinal and aromatic plants. Generally, my role in this work is very little. My friends are special who works in this direction. But I'm happy because we cooperate more than 20 years, Tıbbi State University and Artvin Çoruh Üniversity. She's opened this university, and it's important, our cooperation, because we have only one border and the same nature. So we have to work together, and I'm happy I have a lot of friends. For example, Professor Temalı Uluslararası, we have together projects, master students, doctoral students, and so on. So, presentation title is the ethnomedicinal application of Hypericum species in Georgia. And there are in this my friends, Ingadia Samit, Maria, not Maria Medra, and Nazarnatze. And my name is Kogolitu Batze from Georgia. What about Georgia climate? Georgia varies greatly in climate and relief, and there is sorry. Which one is okay? Can I click? show that. Okay, no, okay. No problem.
Not working. Switch.
This one.
This one in front. This one in front.
No, from. I would like to show. Okay. No problem. And in Georgia, there is Caucasus Mountains. For example, in the north, there is Greater Caucasus, and the Lesser Caucasus in the south. They are joined by a little ridge, which separates Georgia into two parts, and it is a natural barrier that separates Georgia into distinct climate zones. There is Western Georgia, which characterized with humid climate with annual rainfall between from 1,000 to 2,800 millimeter, and it characterized by subtropical conditions in the Black Sea coastal zone. And there is also Eastern Georgia, the second part of Georgia. It characterized by dry continental climate with annual rainfall between from 300 to 600 millimeter. This variation in climatic and topography supports a wide range of ecosystems from subtropical forest to arid zones. Ajaria is saying sometimes Ajaria, sometimes Ajaria, so it is a biodiversity hotspot, but there is hotspot part of Georgian biodiversity hotspot. And Ajaria, located in southwestern Georgia, it is part of the South Caucasus zone. It is known for its rich biodiversity and characterized by subtropical influenced by the Black Sea with mild winters and humid summers. It is home to over 2,500 species of vascular plants, including many endemic species. Recent studies highlighted that the South Caucasus region is especially rich in medicinal plant diversity, preserving traditional knowledge. There are about 184 species of medicinal plants in the region, classified into 68 families and 154 genera. What about ethnomedicine in Ajaria? In Ajaria, plants are the primary source of medicinal products, and ethnomedicine in Ajaria has deep historical roots intertwined with local customs, folklore, and the oral tradition of healing knowledge. Traditional healers played a significant role in treating ailments before modern healthcare became widespread. Local plants play a crucial role in the region's ethnomedicinal practice, deeply embedded in Ajaria's cultural heritage. In the flora of South Caucasus of Ajaria, the genus Hypericum is important to its relict, medicinal, and endemic species, which has been used for treatment by the local population since ancient times. What about preservation of traditional knowledge and modern challenges? I would like to say that in Soviet era, Ajaria's isolation and Georgia's isolation, and with all of countries' republic isolation, helped in Soviet period helped preserve many traditional practices, including the ethnomedicinal use of Hypericum. But there are a lot of challenges, modern challenges, for example, such as habitat loss, climate change, and cultural shifts now strengthen both the biodiversity of Hypericum species and the traditional knowledge associated with their use. And while some medicinal plants are still widely used in rural areas, the loss of traditional healers and lifestyle changes have led to a decline in the transmission of ethnomedicinal knowledge. aim of the study was to analyze the use of the genus Hypericum in Agyara's ethnomedicine, and the study focuses of examines the ethnomedicinal data of Hypericum species found in the Agyara South Colchis region. Another objective was to collect traditional recipes that are widely used and effective in the local population's healing practice. The research employed the traditional road expedition method, and the study focused on the genus Hypericum common in Agyara South Colchis. The research area included from the coastal zone of the Bizi region till mountain areas, especially in regions like Sartikvariatim, Tsvanegomsky, Erge, and Agyaristali. Special attention was given to the south and southwestern steep slopes of the Agyaristali river valley. What about results? Hypericum is included in the state pharmacopoeia of Georgia for its antidiarrheal and antiseptic properties. In medical practice, it is used in the form of a tincture, briquettes, and as part of complex compounds. The plant and its preparations exhibit a wide spectrum of pharmacological activity, including spasmolytic, anti-inflammatory, astringent, antibacterial, P vitamin activity. Commonly used for treatment, cholecystitis, hepatitis, gastritis, colitis, and so on, cystitis, hemorrhoids, stomach, and duodenal ulcers. Hypericum species in Agyara in ethnomedicine, generally there are 5 species there are 3 of them, Hypericum androcerum, and Ferporatum, and Hypericum linearis, and there is their uses in this direction. For example, one of them pro-inflammatory properties and used to treat wound and skin conditions. And second one for respiratory treatments and digestive disorders and so gastritis and ulcers. And Hypericum linearoide used in Georgian folklore as antiseptic properties, often used in traditional receipts and internal and external inflammatory remedies. Hypericum species, there are also 2 species, Hypericum perforatum and Hypericum, which one is in good? Yes. So I tell it is not my direction, sorry. And this one, it's changed. I see now, maybe program change. And there is information about your using antidepressant, anti-inflammatory, and bound healing properties. And the second one is antimicrobial properties, gastrointestinal issues, and so on. So, how would prepare people Hypericum plant material? Harvesting the raw material is the grass, as you know, and you said about it. Harvested during the flowering phase before fruits appears, and the upper part of plant, about 20-25 centimeters long, is carefully cut with a knife, avoiding raw stems. And about drying process, we saw yesterday how they can dry, and there is methodology how people did it and do now some central. The grass is spread, is a thin layer, and dried as well ventilated areas such as an attic. For quicker drying, patching at from 14 to 60 degrees Celsius can be used to speed up the process. It is There are some recipes which collected nitrates in the villages, generally mountain areas, and how to use Hypericum peperoratum for wound healing and skin care, crushed leaves and applied for quick healing of wounds, nodule rashes, nodule and oil severies. And also there is facial care, tampon soaked and in texture and decoction are used to clean the skin. They made alcohol tincture, and there is methodology how they do it, and ointment for joint pain. Also, it used as antiseptic and diuretic, helmintic and diuretic. Also, tuberculosis treatment, stomach disease, and liver disease. This receives nitrate collected in the villages of Mountain Hajara. What about conclusions? There are 5 species, Hypericum. Hypericum are used in medicine, which one they are resourced, and also in ethnomedicine. St. John's wort is used for rapid wound healing, treatment of nodular skin, and for bloody cups during tuberculosis, used to treat jaundice related to liver disease. Common preparation include blood, tincture, and oil dietary from this Hypericum. And recommendations promote sustainable harvesting and conservation of Hypericum species used in traditional medicine, and encourage further research into the medicinal properties of Hypericum species to support their widely use in modern medicine. And they are working now to continue their research in this direction. They have some research in this direction, and they are continue study for us. This research emphasizes the importance of documenting and preserving knowledge about Hypericum species in Hajara. It contributes to a broader understanding of the genus in local ethnomedicine and provides insights into the conservation needs of the plant species and the cultural practices they support. The collected data will help guide future efforts to ensure the sustainability management and conservation of Hypericum species in Hajara, ensuring that their medicinal uses and ecological significance are maintained for future generations. Special thanks for listening to me patiently. Thank you very much.
Any questions?
Thank you. Thank you.
Ladies and gentlemen, wait a bit because of there is updated version will come. It was old version. She just fixed this now. today, no problem. Today we start to try to start a bit early. That's why better maybe if we start at 10 o'clock, we try to, because we have quite a lot of presentation for today. First day we make our partner projects presentation. Second day we did trip, and last day who wanna join our this symposium presentation, and it was too much. That's why we started early. Sorry, but some scientists from Trabzon, yes. I saw. They were welcome. Trabzon is not so far from here. That's why they come today, and they inform us that they have several duty in the university. That's why they can come on for Friday. That's why we also make these changes. Next time, I hope you will make early permission from university and visit our conference from full day, because it was interesting day, 2 day. As I know, or I mistake. we was yesterday in Şavşat area, and did you fight? You didn't fight. Okay, no problem. No problem. What you have?
Sorry.
No problem.
I'm very happy to be standing here. I'm Maria Romanadze, PhD student from Batum State University. My topic is diversity of Hypericum species, Achara, Georgia. We are from ecoregion of the Caucasus. It's a biodiversity hotspot. There are 2 refuge here. It's one of them called Refugium and Hippanian Refugium. The Colchic Refugium includes the Colchic rainforest and wetlands. It's located along the southeastern shores of Black Sea. There is a relict forest. There is a relict forest which has survived the glacial cycle of Ice Age. This place is a refuge for relict and endemic species. There is red lines is borders of refugium. It looks like a shelter for ancient species. There is some ancient species. It looks like jungle on rainforest from Amazon, but it's our forest in Tirana National Parks. There are evergreen shrubs, rhododendrons. It looks like jungles. There are some relict species, not only plants. There is Martensiana caucasica, the salamandra relict species. Also, here is Primula megazerifolia. It's my master thesis about this. There is Rhododendron ponticum, Ilex golfica. It's this plant is evergreen, like herbs and shrubs. You can visit us and look this. The genus Hypericum contains approximately 500 species. Hypericum species can be found all over the world in temperate tropical areas. The genus is most diverse in Turkey. There are 19 species of Hypericum in Georgia. 12 species is common for Achara. Hypericum species are both shrubs and annual and perennial herbs. It is a well-known plant in ethnobotany. The species are rich in phenolic compounds. The most famous species of genus is Hypericum perforatum. It contains hypericin and hyperforin, and it's actively used in pharmacology. This is Acharian species. 12 species. There are Hypericum agaricum. Maybe it's some subspecies and variated from orientally. Hypericum androseamum, Hypericum pisinicum, Hypericum pupleroides, Hypericum caucasicum, Hypericum grossheimi, Hypericum lineroides, montanum, multilum, perforatum, venestum, and Hypericum xylosteiffolium. Some of them are decorative, medicinal, and endemic. My PhD thesis is about Georgia and Turkey transborder species. The most interesting species is Hypericum xylosteiffolium because it's a very rare species. Because it's endemic species for Caucasus ecoregion, it's only split Transcaucasian. And in our country, there are any research, any biochemical research on some. And I'm not a biochemist and biotechnologist. I am plant biodiversity researcher, but cooperate to biotechnologists and biochemistries. In Western Georgian Regional Chromatography Center, we ongoing PHLSPDA analysis of Hypericum xylosteiffolium leaf extracts. We detection for secondary metabolites and identification of chromatogram peaks. There are reveals some important compounds. It's characterized MS, retention time, and wavelength. It's quinic acid, catechin, quercetin, 1-caffeine quinic acid, quercetin-3 or pentosides, synconian LB, antiparasites, and amantoflovin. Maybe I pronunciation it's hard for me. Conclusion: biodiversity of the Achara region is distinguished by Hypericum species, including endemic, medicinal, and decorative ones. Challenge our genus is in-depth research and development in situ and ex situ conservation strategies. Hypericum xylosteiffolium is endemic species in the Caucasus ecoregion. It contains certain secondary metabolites and it's an important plant raw material. There are special thanks for my team. Some of them attend here. Associate Professor Inga Samidze, plant biodiversity researcher. Associated Professor Natala Varshanidze and Doctor of Biology Gia Bolukadze, and staff of Western Georgian Regional Chromatography Center, Professor Aliko Kalandia, Professor Maya Vanidze, and Doctor of Chemistry Jairan Putkaradze. Thanks for you. Thanks for your attention and organization.
Thank you very much. If you have questions. Very good.
Let's continue our second session. We will have 4 nice presentations for this session. 2 from Artvin Çoruh Üniversitesi and 2 from Artvin University. And I would like to invite first PhD student Deniz Gultekin. Deniz, between 2013 2017, he studied at University, Faculty of Engineering and National Sciences, Department of Genetics. In 2017-2020, he completed his master program at Gümüşhane University, Institute of Science and Technology, Department of Biotechnology. In 2021, he started his doctoral education in the Department of Medical Biochemistry at Edin, Technical University, Institute of Health Science.
Synthesis of green nanoparticles from Ruscus Türleri extract and their antioxidant activity. Turkey has a biodiversity that is not found in many other countries due to its location at the intersection of 2 important gas centers, the Mediterranean and the Middle East, and 3 main biogeographic regions. There are different bioclimatic types, topographic, geographic, and pharmacologic sectors. Soil diversity, different geographic conditions, the plants of different types, such as sea, lakes, river, salt and saline lakes, and different ecosystem types. For this region, about 12,000 taxa belong to different families, and genera are naturally distributed in Turkey. Considering that there are 12,500 plant species in the anti-European continent, and the area of the European continent is about 15 times larger than the area of our country. It's clear that the Anatolian flora has a very important place in the world. However, experimentally, 3,500 of these 12,000 plant taxa are endemic species. In recent years, serious side effects caused by the use of synthetic drugs and economic and medical problems caused by these drugs and might treatment with plants popular again. Today, the increase in interest in organic and natural product foods has also increased the tendency toward medical and aromatic plants. One of these plants is Ruscus Türleri. It is located in the Earth Black Sea region of our country. This is success extraction. Extracts in ethanol 2 hours at 210 degrees by success extraction. Ruscus Türleri leaf ethanol extract. Ruscus Türleri rhizome ethanol extract. The antioxidant properties of leaf and rhizome extracts were investigated used for different antioxidant methods. These were DPPH, FRAP, total polyphenol, total flavonoids. When leaf and rhizome extracts are compared, the total flavonoid amount are experimentally 41 milligrams of quercetin per gram of sample in the leaf. Rhizome 1.62 grams quercetin per gram sample. Comparing leaf and rhizome extracts, total polyphenol amounts were 27.32 milligrams of gallic acid per grams of sample. Rhizome 8.42 milligrams gallic acid per gram sample. When leaf and rhizome extracts were compared, DPPH IC50 amounts were 544 milligrams per milliliter in the leaf. DPPH IC50 amounts of rhizome 459 milligrams per milliliter. Leaf and rhizome extracts PRAP values 66.41 milligrams of Turlops equivalent per grams of sample. Rhizome 26.6 milligrams Turlops equivalent divided gram sample. Nanoparticle synthesis. Nanoparticles are very tiny material that have size between 100 nanometer. Their small size enable them to penetrate into the cell very easily. This picture first water extract 2 gram sample were boiled at 125 degrees 60 minutes. The copper chloride was dissolved in the water, and when it reached a certain temperature, the plant extract was slowly added to solution, and this process was continued until a color change was observed. Then washed with dissolved water, and finally washed with ethanol to obtain nanoparticles. As shown figure, UV spectrum of copper nanoparticles, the maximum absorbance peak occur at about 300 nanometer, which was attributed to the absorbance band copper oxide nanoparticle. The absorbance band of copper oxide nanoparticle at 3 nanometer confirmed the formation of copper of nanoparticle. As the volume increasing, the absorbance intensity decrease indicated that the formation did not occur at the large volume. Copper oxide nanoparticles were formed in the agreement with studies suggest that copper oxide nanoparticles formed between 200 and 250 nanometers. FTR spectrum, copper nanoparticles. The functional group present in the water extracts of Ruscus colchicus and copper oxide nanoparticles were identified using FTR spectrophotometry in the range of 4,400 peak. The characteristic peak at the 500 and 5 peak confirming the formation of copper oxide nanoparticle indicates the formation of copper oxide stranging vibration. Therefore, from the FTR analysis, it can be started that polychemicals such as polyphenol and flavonoids present in the water extract are responsible for reduction and stabilizing metal ions to form copper oxide nanoparticles. Iron nanoparticle synthesis. Nanoparticle synthesis were carried out according to green synthesis methods, minor modification. First of all, iron sulfate heptahydride salt in the molar ratio of 1:1 were dissolved in 10 milliliter of dissolved water and mixed for 60 minutes. at 125 degrees after incubation. The resulting mixture was collated to room temperature. After synthesis, the supernatant was removed. The resulting principle was washing first with distilled water and then with ethanol and dry at room temperature. UV spectrum of iron nanoparticles, as shown in figure, the maximum absorbance peak occurred at about 219, which was attributed to absorbance band iron oxide nanoparticle. The absorption band of iron oxide nanoparticle at 219 to 320 nanometer confirmed the formation of iron oxide nanoparticle. As the wiring increased to absorbance intensity decreased, indicated that the formation did not occur at a large wave. Functional group present in the water extract of Ruscus and iron oxide nanoparticle were identified using FTR spectrophotometry in the range of 665 at point. The characteristic pit at 505 peak confirming the formation of iron oxide nanoparticles indicated the formation of iron and oxide staging vibration. Therefore, from the FTR analysis, it can be concluded that photochemical such as polyphenol and flavonoids present in the water extract are responsible for reducing and stabilizing metal ions to form iron oxide nanoparticle. Two nanoparticle DPPH comparison. A DPPH comparison of the nanoparticle obtained was performed. Iron nanoparticles were found to have bigger antioxidant capacity. Thank you.
Thank you very much. Please, questions? I will ask you one question. Is like this, you are obtaining copper oxide and iron or metallic iron. All chemical with synthesis of copper.
Yes. Bakır oksit, copper oksit, yes. The yazmamış. Yes.
It's different. It's still on progress with the some images.
Yes.
Questions? Thank you very much.
I want to say thank you for this, guys, because of this is the application of plants or plants extraction. Thank you for your effort.
Next presentation from same university, from Artvin Çoruh Üniversitesi, and presenter is Ahmet Özkan. The title of the presentation is Synthesis of Iron Oxide Nanoparticles from Hypericum Psilofolium Extract. Ahmed is lecturer at Medical Aromatic Plant Application and Research Center. Department is in biology. PhD thesis was about targeting some genes for treatment of lung cancer via serum-loaded non-viral vectors. Working area are drug delivery, liposomes, magnetic nanoparticles, anti-cancer, antimicrobial, antioxidant, and metabolicity activities. Please, you have 25 minutes. Keep 5 minutes for questions.
Yeah, thank you, Chair, and yes, thank you for attention. We work on synthesis of iron oxide nanoparticles from Hypericum psilocybe extract. I'm one of the scientists research of 4 researchers. Nanoparticles are a broad class of materials containing particulate matter with a size at least less than 100 nanometer. They can be 0D, 1D, 2D, or 3-dimensional depending on their overall shape. Nanoparticles are generally divided into several categories based on their morphology, size, and chemical properties. Nanoparticles show enhanced properties due to their size, morphology, and distribution, and widely used in various scientific fields. Biological materials such as microorganisms and plants are less costly and environmentally friendly. Sorry, I take the pointer. As we see, nanoparticles are between 1 nanometer to 100 nanometer size. They are very tiny, and their most important advantages they can be easily penetrate into cell, and it can show activity. Especially metal oxides are very important to their biological activities. As we see, there are 2 routes of synthesized nanoparticles from top to down approach and bottom up approach. Bottom up approach are categorized into 2 different routes, chemical and biological. In biological plants, route, plants also used as microorganisms based algae, bacteria, fungi, and yeast. We are focused on plant material based synthesis. Nowadays, green synthesis techniques for nanoparticles are recommended conventional synthesis techniques as they have several advantages in terms of cost, energy utilization, non-toxicity, and safety. Green synthesis is defined as the application of biologically friendly elements such as plants, bacteria, and fungi for their synthesis of nanoparticles. Green synthesis of iron nanoparticles is reliable, sustainable, and eco-friendly protocol for synthesized nanoparticles. There are also there are plenty of plants in nature and have pharmaceutical significance. So we have a very rich sources. Nanoparticles, we mentioned before, there are 3 ways to synthesize. We are focused on green synthesis with plants. So this symposium is about Hypericum and Ruscus species. So we want to use plants to synthesize nanoparticles. Sorry. With plants, I'm sorry. Okay. Yes. In plant parts, you can use leaves, root, flower, and fruit to synthesize nanoparticles. The plant extract work as both reducing and coating agents in the synthesis of nanoparticles. So our aim is to synthesize iron oxide nanoparticles via green synthesis method and test biological activities of these nanoparticles. For this goal, firstly, Hypericum xylostefolium was extracted, and then iron nanoparticles synthesized, and then the antioxidant activity was tested. Nanoparticles was carried out according to the green synthesis method via minor modifications. Firstly, we use iron sulfate in molar ratio, and then we dissolved in distilled water, then mixed for 60 minutes at 125 Celsius degree. After incubation, the resulting mixture was cooled to room temperature. After synthesized, the supernatant was removed. The resulting precipitate was washed firstly with distilled water and then with ethanol and dried at room temperature. The characterization was carried out with FTR and UV-Vis analysis. The antioxidant activity was tested due to the pPH with minor modification. The experiment was applied in triplicate. Ethanol was used as blank, and free radical was tested of nanoparticle samples to determine the scavenging activities. So we used the equation in here. As a result, we can see the peak at 646 in FTR result of iron oxide nanoparticles. UV spectrum result was showed at 290 nanometer. There's a peak according to the literature. We can see the figures, pictures of firstly the synthesis. Firstly we mixed and heated to 125 degrees, and then we added the Hypericum extract, aqueous extract, and then we incubated for 60 minutes, and then we washed and centrifuged. After that, we dried at room temperature, and we can see the precipitation in here. So then we tested as antioxidant activity, and we can see the iron oxide nanoparticles was scavenging the pPH against to control. In the literature, it is stated that iron oxide nanoparticles are formed when a color change and is observed during the reaction. In our study, we also see precipitation and the color change. In addition, some peaks disappeared in the FTR analysis, and UV-Vis absorbance values also showed the presence of iron nanoparticles. It was observed that nanoparticles synthesized with Hypericum leaves have antioxidant activity. Antioxidant activity showed at concentration 62.5 microgram in milliliter, and it was scavenging percentage of 73, approximately 73%. Natural extracts and biological components obtained from organisms such as plants, bacteria, seaweed, algae, fungi, etc. are used to synthesize nanoparticles. The green synthesis is a safer and more effective method to obtain nanoparticles. The most important advantage of green synthesis of nanoparticles are simple method, low cost, and without toxicity. Green synthesis is one of the two synthesis methods and is called bottom-up synthesis. In bottom-up synthesis, atoms are collected and formed into clusters to form nanomaterials. Medicinal plants have been used to treat disease since the beginning of humanity. In the antioxidant activity study of green synthesized iron oxide nanoparticles, it was stated that free radical scavenging activity percentage increased with increasing iron oxide nanoparticle concentration. The high concentration of leaf extract enhanced the phytochemical content of the extract, which reduced the precursors quickly, leading to rapid nanoparticle formation that enhanced the absorbance value. For this reason, in future studies, the green synthesized metal nanoparticles will be synthesized and tested for some other biological activities. This study was supported by Artvin Çoruh Üniversitesi Scientific Research Projects Coordination Unit, and the project number was this. And here are the references. And I'm really thankful to organization committee and you and the chair of this green chemistry symposium.
Thank you very much. Very interesting presentation. Please ask this.
You mentioned questions about size of your nanoparticles. You said that you exactly have the nanoparticles.
Yes.
But do you have any CM or was it
it's still on progress to take same image and XRD analysis and also DLC analysis to find out the nano size of these materials.
Yes, and it's good also to obtain the loss of diffraction and you will obtain the information about size distribution.
Yes, maybe in next in future. Yeah, thank you. Thank you very much.
Next question.
Another comment also how if they can clog again each other or compare names as well.
Yes.
I had the same question. About stability.
Stability. We just dissolve in water, and we will take some DRC analysis to find out the stability of these nanoparticles. We also will test the time degradation. Okay.
Then is a matter of more curiosity to say why use synthesis instead of preparation? Because the concept of synthesis should be a bit more inorganic synthesis instead of preparation.
I'm
in the literature you find also synthesis. But is the proper or improper use of the term of synthesis?
Yes, it's still on progress. Yeah.
The stability of nanoparticles is much more good than the synthesis.
Yes, we will encapsulate these nanoparticles into liposomes, and we will test against cancer cells. Still in the future.
To keep nanoparticles without some formation itself in the water, it will be very interesting.
Yeah. Thank you.
Next question. Okay. Stop.
Thank you.
Okay, next presentation from Artvin University, Associate Professor Yeşil. If you want, the title of presentation is Ethnomedicine and Application of Hypericum Species in the investigation of antimicrobial effects of secondary metabolisms from Ruscus against some human pathogens. about short biodata about our presenter. She is graduated from Karadeniz Technical University Faculty of Science, Department in Biology, with doctorate in microbiology and biology. She is now assistant professor at Artvin University, Toya Vocation School, Department of Medical Services and Techniques. She has been participating in 11 research projects from European Union, Tıbbi, Target, EAP, and some public projects. She is author of 20 articles and 45 papers published at international and local national journals. She has age index of 6, and she has this research area is the creation of microinsecticides derived from EBFs and their biological control, and antimicrobial properties of toxins and secondary metabolites, which are products of microorganisms, such as bacteria and fungi.
Oh, thank you. Thank you.
So you have 25 minutes.
Okay.
Please keep 5 minutes for questions.
Okay. Dear participants, welcome to my presentation. I would like to talk about investigation of the antimicrobial effects of secondary metabolites from Ruscus aculeatus against some human pathogens. First of all, I will give short information about these plants. The plant Ruscus aculeatus is of the great importance due to its numerous applications in medicine, industry, and horticulture, and including its uses and ornamental plants. And this plant is most common habitat on Earth, generally in northern, southern Europe, Anatolia, and North Africa. This plant does not grow in cold areas because it is resistant to cold, it's not durable, and grow in almost all seasons. The plant is mostly grown in the Mediterranean, Black Sea, and Marmara region in Turkey. This plant is used as the diuretic and anti-infective agent in the treatment varicose veins and hemorrhoids, as well as to prevent vascular occlusion and circulatory disorders. Studies have found the bacteriostatic effects of Ruscus aculeatus on Staphylococcus aureus is similar to the effect of antibiotics. Rutin hydrat and p-coumaric acid synthesized from plant extract are found to be effective in some human bacterial pathogens, and it was found to be higher than streptomycin and especially ampicillin. This study was conducted to determine the antimicrobial properties of extracts obtained from the leaves and stems of Ruscus aculeatus against a range of human pathogenic bacteria and fungal pathogens in two different solvents. These solvents, ethanol and methanol. First of all, Ruscus aculeatus plant was collected from Yalıncak, Trabzon in May 2024. And as you can see this photograph, the leaves separate from the branch and treated with the solvent, ethanol and methanol, for each application part and keep in the dark environment for 7 days. This is the classical method. And following 7 days of filtration, the solution was transferred into the volumetric flakes. After this process, the mixtures obtained were separated from ethanol and methanol, used as solvent and evaporated for purification. They were soaked with the MCO and stored in the dark glass bottle at 4 centigrade degrees. As you can see that the antimicrobial activity, experiment diffusion method, you can see the bacterial and fungal strain used in the study on the slide. And these bacteria and fungi were sprayed in the appropriate media, and secondary metabolite obtained were applied in the amount given on the slide. And then the results were evaluated with positive and negative control. As you can see, we use a negative control DMCO and kanamycin for bacteria or fluconazole for fungi is positive control. Then, after incubation, after 24 and 48 hours, the diameter zone, as you can see, formed around the disc were measured. As you can see, that this FF photograph as experiment, and we inhibition zone, we evaluated our result using SPSS program. And the results table. Firstly, the analysis of ES column-related compound demonstrated no actional against Enterobacter cholerae, Staphylococcus epidermis, Klebsiella pneumonia, and Candida albicans, as you can see. However, it was observed to form a significant inhibition zone in other bacterial groups, as you can see. The examination of the EL column revealed that it was ineffective against Salmonella typhimurium, as you can, Salmonella typhimurium, and Staphylococcus epidermis, Klebsiella pneumonia, and Candida albicans. However, it demonstrated greater efficacy against Bacillus subtilis compared to the other strain tested. The result demonstrated that the secondary metabolites derived from the stem exhibited greater efficacy than the leaf extract, with an inhibition zone of 11 mm against Escherichia coli, Staphylococcus aureus, and Salmonella typhimurium. The other result, EMC clones, was found to be ineffective against Enterobacter cholerae, Staphylococcus aureus, Staphylococcus epidermidis, and Klebsiella pneumonia. However, it demonstrated a notable effective against Bacillus subtilis with a recorded zone diameter of 16.45 mm, as you can see this table. An examination of ML clones reveals that it has been demonstrated to be effective against Enterobacter cholerae, Bacillus subtilis, and Enterobacter faecalis. The other result, the comprehensive examination of the data revealed that all metabolites demonstrated efficacy against the green line, Bacillus subtilis and Enterobacter faecalis, while only MC exhibited activity against Candida albicans. It was determined that ES column extraction was effective against Salmonella typhimurium. Lastly, in particular, the height zone created by MC on Bacillus subtilis in comparison to other strains indicate that secondary metabolites derived from this plant may be potential application, particularly in treatment of infection causes by Bacillus subtilis. Thank you for listening.
Thank you very much. These questions?
Thank you for your presentation. And
You're welcome.
Some questions. What kind of microorganisms
not clinical.
Yes, this strains is EFCC strains, but this method used this article and the other studies, and we can use these strains.
Yes, and the second question, did you determine minimal inhibition?
Yeah, we will claim to minimal in MEK concentration, but not didn't work. Okay.
Yes, I understand.
Okay.
Thank you.
Thank you. So, thank you very much for this presentation. Ruscus also maybe you know also in Artvin, especially
Yes.
Center, Çoruh Üniversitesi,
Yeah. Yeah.
Too much. There is too much everywhere. Have you ever planned also to collect from Artvin region and compare with your region? Do you have any plan like this?
Yes, I like. We plan to collected plants. Maybe. Okay, thank you.
Last presentation of this session is our scientist from Temalı Uluslararası Sempozyuma, Assistant Professor Usta, and the title of presentation is Investigation of Antimicrobial Effects of Oil Extraction from the Fruit of Ruscus aculeatus. Short biodata about her. She completed her PhD in microbiology at Karadeniz Technical University, Department of Biology in 2017. In 2018, he started to work as an assistant professor at Artvin Çoruh Üniversitesi. After 2 years, she moved to Trabzon University and take a position of assistant professor. She is a participant of 10 projects from the European Union, Tıbbi and BAP. She is author of 17 articles and 20 papers and 6 books chapters. She's research interests focus on bee health, bee disease, and antimicrobial effects of different products. So you are bee doctor. Okay.
Thank you for your nice presentation. Welcome. Today I want to mention about investigation of antimicrobial effects of oil extraction from the fruit of Ruscus. First of all, I want to say that in our work team, Dr. Bizi and me are working on microbiology, and the other members are working on plants. And this is a part of, also small part of a study. So the results are not clear, I can say, and the study is also working, continuing. On previous presentation, Dr. Bizi also mentioned about Ruscus, and I want to say a bit about Ruscus, want to talk not more. Ruscus, commonly we know Bacchus broom, because it's known as Bacchus broom because it was used as especially by Bacchus in the Middle Age. Bacchus used these branches of this plant to clean the meat counters. This plant grows naturally in the Mediterranean region, south and west of Europe, North Africa and West Asia. Ruscus aculeatus is a woody shrub recognized by its small spiny leaves and, as you see on the photo, bright red berries. It includes steroidal saponins, ruscogenin and essential oils especially, flavonoids, resin and minerals. The underground parts, also roots, of this plant have a long traditional use. In Palestine's folk medicine, rhizome extract was used against skin disease, while in central Italy it's used in the treatment of warts and chilblains. In Turkey, its decoction was used internally towards eczema and kidney stones and nephrites. In some parts of Italy, the same preparation was used with colics. It's also used locally against inflammation and for arthritis. Essential oils are categorized under four main headings according to the way they are isolated from plants. These methods are cold extraction, distillation, supercritical carbon dioxide extraction, and enflorage methods. The most efficient active substance different methods have been developed in order to obtain the oils in a light proof glass, and it's recommended to store them in bottles at 4 centigrade degrees. Plants containing etheric oil are rich in bioactive compounds such as alkaloids, flavonoids, and terpenoids, and also in the control against disease, especially antiviral, antimicrobial, and antifungal and pests, the oils are used. Due to the fact that their main components are terpenes, essential oils are widely used in many fields. And all of these importance for this Ruscus, what our aim? This study was an investigation of antimicrobial effects of oil extraction from the fruit of Ruscus against a range of human pathogenic bacterial and fungal pathogen in and hexan. In the literature, antimicrobial effects has been investigated using different solvents. Also, we will try these different solvents, but it was considered to investigate the antimicrobial properties of a local sample using N-hexan. For this aim, plant was collected from Trabzon in May. The ripe fruits of Ruscus plant were plucked from the branch and dried in an oven at 40 centigrade degrees for 1 day. At the end of the period, the dry weight was weighed and crushed with a mortar and transferred to a socklet cartridge. After 2050 milliliter N-hexan was added to the 500 milliliter flask of the derris, it was evaporated at temperature program number 1. After oil extraction from the fruits in the cartridge 3 times in about 6 hours, the derris was switched off and the oil and hexan mixture in the flask was taken to the rotary evaporator derris. Evaporation was performed at 40 centigrade degrees, 80 rpm, and the volume of oil remaining after evaporation of N-hexan was calculated. After that, the extract obtained was used for antimicrobial tests. Pathogens, as you can see here, ATCC strains we used. Pathogens were prepared in accordance with the principles of diffusion methods, and discs, sterile discs were placed after sowing on medya. Medya is Müller Hinton agar. After 24 or 47 hours, the diameters of the zones formed around the discs were measured. At the end of the antimicrobial study, it was determined that the extraction was effective on Bacillus subtilis only. Bacillus subtilis is caused by eye illness, and statistical analyses were done. Only one positive result for in this short experiment. So in the beginning I mentioned these are not clear results. It's also continuing. This study is also continuing. So now only for these small results we can say maybe or it has potential for use eye illness, fruits oil. Only we can say this limited result maybe. So I want to thank Rasim Öztürk for providing and identifying the plants he had us, and also we would like to thank the Trabzon Regional Directorate, and also thank for Dr. Biryol and Ali Soydinç, and thank you for your listening.
Thank you very much.